Okay, welcome everyone to this joint seminar of the Society for Experimental Biology and the Journal of Experimental Botany. My name is Rainer Meltzer. I'm an assistant professor at University College Dublin, and it's my pleasure today to introduce Daphne Goring as our speaker. So Daphne studied at McGill University in Canada, and she then went to the University of Toronto, where she did her PhD. And now she is a professor at the University of Toronto at the Department of Cell and Systems Biology. And well, I'd say she works on the best study objects of all. She's working on flowers. And in her work, she analyzes one of the most fundamental questions, and that is how do plants distinguish self from non-self? And to study that, she uses the interaction between pollen and pistil. So I don't want to talk too much about Daphne's research because I think she's going to talk about that. But suffice it to say that her research is so outstanding that she was named a fellow of the American Association for the Advancement of Science in 2011, I believe. So Daphne is also very active in promoting science and in serving the scientific community. So she's the president of the Canadian Society of Plant Biology, and she is also an associate editor for the Journal of Experimental Botany. So I'm looking forward to Daphne's talk. And before we start, I have one small technical remark. So we try to create an inclusive atmosphere, and it can sometimes be a little bit daunting for students and postdocs to ask questions. And to try to mitigate that, we would ask all senior PIs in the audience maybe to wait 1 or 2 minutes after the talk before they ask a question so that we can give students and postdocs the opportunity. Okay, so without further ado, I'm handing over to you, Daphne.
Thank you, Rainer, for that very nice introduction. So thank you to SCB and the Journal of Experimental Botany for inviting me to give this presentation today. And I'm going to talk about these two groups of receptor-like kinases that are involved in pollen-pistil interactions. So this is work that was recently published in the Journal of Experimental Botany. And towards the end of this presentation, I am going to talk a little bit about where we are going now with this project. Okay, so in my research group, we're interested in studying pollen-pistil interactions in the Brassicaceae family. So that includes Arabidopsis thaliana, which is the topic of the presentation today. And we look at both the self-incompatibility pathway, where you get the rejection of self-incompatible pollen, as well as the compatible pollen response pathway, which is what I'm going to be talking about today. So over here, you can see an image of an Arabidopsis thaliana flower that's been pollinated. So the male pollen grains from the anther have been released, and they're landing on the top of the pistil. So they land on top of the stigma, which is at the top tip of the pistil. And these pollen grains are going to come in contact with these finger-like cells called stigmatic papillae. So you can see that in this diagram here, where you've got a pollen grain contacting one of these stigmatic papillae. And this is basically the first checkpoint in this pathway that allows for compatible pollen to be accepted. And basically, there's a pollen discrimination system involved that would allow for the recognition of compatible pollen. And the first thing that occurs is this pollen is going to hydrate, and this is due to water being released from the stigmatic papillae. So what are the features of the Brassicaceae family is that they have what are called dry stigmas, and they regulate the release of water for pollen hydration. So it's only when the pollen has been recognized as compatible that this occurs. And the reason why you need pollen grains to hydrate is when they first arrive, they're desiccated and metabolically inactive. So the first step is for water to be released for the compatible pollen grain to hydrate, and then it's going to germinate and form a pollen tube. And this pollen tube is going to grow through the cell wall of the stigmatic papilla to the underlying tissue. So the next step is that you get pollen tube growth through the stigma, the style, and into the transmitting tract. And at this point, the pollen tubes are actually growing between the cells of the tissue in this region. Okay, and so there's several checkpoints along this way to the pollen tube reaching an ovule, and these include pollen tube guidance to the ovule, pollen tube reception at the ovule, and the prevention of polyspermy. And what all of this really means is that it's about guiding these pollen tubes, a pollen tube to an ovule where it can release the sperm cells for fertilization. And so the next step is that you end up with fertilization and seed development. So that's the end point of this whole process. Okay, so we actually know a lot about the players that are involved in the system, and this is just a generic cartoon of this. We know that there's a number of peptide ligands that are involved, and they're perceived by cell surface receptors. In this case, it's receptor kinases. And these receptor kinases then activate intracellular signaling pathways and cellular responses that enable these successful pollen-pistil interactions. So I'm not going to talk about all the different players that we know about. I'm just going to focus on the very beginning step, which is the extracellular signals and the receptor kinases. And I'll review what is known about these players in pollen-pistil interactions so I can provide some context for the discovery that we made. Okay, so very recently, Liu et al. Published a paper where they identified two receptor kinases, Feronia and Angia, which set up a negative regulatory loop to prevent pollen hydration. So they're involved in this autocrine signaling pathway with this Ralph33 peptide, which results in the production of ROS. And the authors showed that the ROS that's in these stigmatic papillae prevent pollen hydration. So in order for the compatible pollen grain to hydrate, it needs to overcome this ROS production. And what the authors found was that these PCBs were involved. So previous work by Wang et al. Showed that these PCBs are small peptide-like ligands that are required for pollen hydration. And so the PCBs were found to bind to Feronia Angia and shut down the ROS production to allow for pollen hydration. So we think there's additional players involved in a positive regulatory loop to allow for pollen hydration, and I'll talk about that more later. Okay, so we actually don't know a lot about the in-between stages of what are the molecular players regulating pollen-pistil interactions in the stigma and the style. But towards the end, we do know quite a bit about the pathways that are involved for pollen tube guidance and pollen tube reception and rupture. So I'm only showing you the ligands and the receptor kinases that are involved. There are a lot of other players that are also involved at this stage. So we have receptor kinases on the pollen tube that perceive signals, peptide ligands secreted by the synergid cells to lure this pollen tube to an unfertilized ovule. And when it reaches the ovule, the next step that has to happen is that pollen tube has to burst so that it can release the sperm cells. And that involves additional receptor kinases, both of them pollen tube and on the synergid cells, as well as peptide ligands, that essentially ensure that this pollen tube doesn't rupture too early, that it keeps growing until it reaches a stage and then it ruptures to release the sperm cells. Okay, so as I mentioned, we don't really know about much about the players that are regulating the pollen-pistil interactions in the earlier stages. So the question we asked is, are there receptor kinases perceiving compatible pollen grains and pollen tubes in the upper part of the pistil? We know that there are no stigma-specific receptor kinase genes when we look through all the transcriptome data sets, but we did identify a stigma-specific pseudokinase gene called brassicin-1. And brassicin-1 encodes a kinase domain, but it's called a pseudokinase because it's lacking the catalytic residues that would allow it to phosphorylate substrates or itself. So it's called a pseudokinase. This is a promoter GUS analysis here showing the BCAN1 promoter driving GUS expression only in the stigma. And so this confirms that it is a stigma-specific gene. And the protein also has a myristoylation site at the N-terminus, which would allow it to localize to the plasma membrane. So we actually don't really know what BCAN1 is doing in the stigma. It's a bit of a complicated story, and I don't have time to go into it today. But we decided to take a leap of faith and see if we could use it as a way to identify candidate receptor kinases that are functioning in the stigma. And the reason why we did that is we know that cytoplasmic kinases and pseudokinases are found in complexes with receptor kinases. So we hypothesized that BCAN1 may be interacting with receptor kinases in the stigma, and that may help us find some candidates for further testing. Okay, so why do we have to do that? Well, it turns out there's a lot of receptor kinase genes in the Arabidopsis genome, over 400, and many of them are expressed in the stigma. So it'd be really difficult to just go through and randomly knock them out to see if we can see a phenotype. So we needed a more a strategy to try and reduce the number of candidates to test. And so we use Arabidopsis thaliana and Arabidopsis lyrata BCAN1 clones in a yeast 2 hybrid screen to discover interacting receptor kinases as candidates for further testing. So for the 2 hybrid screen, we looked at a number of different stigma data sets to pick out candidates to test. And we then did pairwise interactions between those candidates and BCAN1. So we are essentially cloning the cytosolic kinase domains from these receptor kinases and testing their ability to interact with BCAN1. We use mostly LR receptor kinases, and that's this middle group here, because they make up over 200 of the predicted receptor kinase genes in Arabidopsis. And there are a lot of resources for them, including cDNA collection of gateway entry clones, which made their cloning very simple. Also picked out a few other RKs, receptor kinases from other classes, and cloned their cytosolic kinase domains as well. Okay, and positive interactions were then confirmed by biomolecular fluorescence complementation, BIFC. I'm not going to show you this today because I want to move on to the other part after I show you the 2 hybrid screen, which is to look for mutant phenotypes. So for the interactors, we found gene knockout lines and looked to see if they had altered pollen pistil interaction phenotypes. Okay, so this is an example of the 2 hybrid screen and the results that we saw. There are close to 60 different receptor kinases that were tested against the BCAN1s. It turned out that Arabidopsis lyrata BCAN1 was the clone that interacted with the cytosolic kinase domains belonging to the RKF1 and RKFL1 group from different, and BCAN1 was able to interact with different Brassicaceae clones for RKF1 and RKFL1. And we also found interactions with SERC1 and SERC3. So this is the key for this diagram, showing you which receptor kinases were tested. Every time you see a blue, that means there is a positive interaction between BCAN1 and that receptor kinase. So you can see the RKF1, RKFL1, and some of the Brassicaceae homologs interacted, as well as SERC1 and SERC3. So you can see the blue dots there. Okay. So these positive interactors belong to 2 groups of receptor kinases, the LR8-2 family for RKF1 and RKFL1, and the LRR2 subfamily for SERC1 and SERC3, which is also known as BAC1. And so what are these two groups? Well, I'm going to start with Circ1 and Circ3. These are well-known receptor kinases. They're part of the Circ family, and they function as co-receptors for a number of other LR receptor kinases. So the Circs have really small extracellular domains with just a few leucine-rich repeats, and they act as co-receptors with other receptor kinases that have much larger extracellular domains with a lot more leucine-rich repeats. So the example I've shown here is BRY1. So that's one group of the interactors. And the other group was the RKF1 and RKFL1, which belong to the LR8-2 subfamily of receptor kinases. So this is an interesting family because they have this hybrid extracellular domain where they have a group of leucine-rich repeats at the end terminus, followed by a molectin domain. And this molectin domain is also found in this well-known receptor kinase Feronia, which is involved in a couple of different stages of pollen pistil interactions, which I showed at the beginning of my introduction. And Feronia has a number of other roles as well. Okay, so we actually don't know a lot about these LR8-2 members. There's some papers on them, but their functions are still fairly elusive. And in the case of RKF1, it was called RKF1, which stands for receptor kinase flower 1, because it's highly expressed in flowers, but there's nothing else known about it. And we named the other three members here because, as I'll show you in a couple of slides, they're all found as a cluster, and they're the most closely related paralogs to each other. So we named them RKFL1, 2, and 3 for receptor like kinase flower like 1, 2, and 3. Okay, so the fact that BKM1 interacted with the Circs and the RKF1, RKFL1 clones raised the idea that maybe they could potentially function in a complex like we know for Circs and other co-receptors. I can't shed any light on that today, but that certainly is one of the intriguing questions that we've been thinking about. So in terms of studying their function, we want to first make sure that they really are expressed where we want them to be, which is in the stigma. And this is a heat map from the TRAVA data set showing you that RKF1 and RKFL1 do show good expression in the stigma, as well as Circ1 and Circ3. And you can see that they're expressed in other tissues as well. So it turns out these receptor kinase genes are expressed in a large number of tissues, but that does include the stigma. And down here is data from a LRRK promoter GUS study. So Wu et al. Basically cloned promoters from over 200 leucine-rich repeat receptor kinase genes and fused them to GUS to show where they're expressed. And so we were able to look at this data to see that RKF1 is expressed nicely in the stigma and the style of the pistil. RKFL1 in this promoter GUS study shows a little bit less expression, but we do know it's expressed in the stigma. Circ1 and Circ3 also show good expression in the stigma and style, so in the upper part of the pistil that you can see here. At the very end of the talk, I'm going to bring up some of these genes. So these are other LR8-2 kinase members, and you can see that their promoters also drive strong expression in the upper part of the pistil, in the stigma and the style. Okay, so to further explore their potential functions in pollen pistil interactions, we identified mutants for these different interacting receptor kinase genes. And so we have tDNA knockouts for RKF1 and RKFL1. We use well-known tDNA knockout lines for Circ1 and Circ3, which are Circ1-1 and BAC1-4. Now, as I mentioned, RKF1 and RKFL1 are in this cluster of 4 receptor kinase genes. And these are, as I mentioned, the closest related paralogs to each other. So in order to make a higher order mutant, we ended up making a CRISPR deletion that deleted this entire region. So it's a 25 kilobase cluster deletion, removing all 4 of these genes. And we call this RKF delta. So for the assays to explore their function, remember we're focused on the pistil side of this equation. We took wild type Columbia pollen grains and then pollinated wild type and mutant pistils to look for phenotypes. So the phenotypes I'm going to be telling you about are pollen phenotypes, but remember that the pollen is wild type, and so what it really is the ability of these mutant pistils to be able to support or not support these wild type pollen grains. So the first assay that I'll show you is called the pollen hydration assay. This is a very simple assay where we measure the diameter of the pollen at 0 and 10 minutes. So the desiccated pollen grain has this oval shape, and when it hydrates, it becomes rounder, and you can measure this diameter increase as a proxy for hydration. And then the next two assays that I'm also going to show you look at pollen tube growth. So that involves aniline blue stain of callus deposits in the pollen tubes, as well as using a Gus marker pollen, which we can then stain for Gus activity to visualize the pollen tubes. Okay, so these mutants, it's very important to make sure that the pistils are normal because we don't want to have developmental phenotypes when we're trying to understand how they're functioning in pollen-pistil interactions. So here's an image of these flowers where we've moved the petals so that you can clearly see the pistils in the middle. And you can see that disrupting these different receptor kinase genes has no impact on the pistils. They're all wild type in appearance. The only phenotype we noticed was that Circ1 BAC1 causes a reduced elongation of the stamen filaments, but because we're focused on the pistils and always using wild type pollen, this is not an issue for our assays. And here are images of the plants. So you can see that when we delete all 4 receptor kinase genes in the RKF delta mutation, these plants still remain completely wild type in appearance. They look the same as Columbia. And the only phenotypes at this stage are brought in by Circ1 BAC1 because they're involved in steroid signaling, and so you see this reduced growth phenotype. But they are still able to produce these wild type looking pistils. Okay, so the pollen hydration assay. So remember, we're adding wild type pollen on wild type and mutant pistils and measuring the increase in diameter at 10 minutes. So this bottom row of data here is the 0 time point for Columbia pollen on the Columbia pollen pistils and all the different mutant pistils. And you can see that there's no changes, which is what you would expect at 0 minutes. The top row of data is the measurements for 10 minutes. So you can see that when we mutate RKF1 or RKFL1, there's no change in the pollen hydration at 10 minutes. It's the same as wild type Columbia. So these mutant pistils are supporting the same level of hydration as wild type Columbia pistils. Same for Circ1 BAC1. It does not, these mutant pistils are also able to support wild type pollen hydration levels. But when we deleted the RKF cluster, we now see this reduced level of hydration at 10 minutes. So remember, this is wild type pollen on the mutant pistils, and so now these pollen grains aren't able to hydrate as efficiently by 10 minutes compared to Columbia and the Circ1 BAC1 mutant. And when we combine it with the Circ1 BAC1 mutation, it's still the same level. And so this tells us that the RKF cluster is responsible for this pollen hydration defect. We were able to rescue this defect by expressing the RKF1 cDNA under the control of the BCAN1 stigma promoter. And we have 2 lines, 3 and 8, that have good expression of RKF1, and they're able to rescue this decreased pollen hydration back up to wild type levels seen for Columbia. We have 1 line, number 4, which doesn't express the transgene very well, and it is not able to rescue this defect. So this is a nice control to show us that we really are focused on the RKF cluster and their role in pollen hydration. All right, so here's a set of aniline blue stains to look at pollen tube growth. The top row are bright field images where you can see all the pollen grains contacting the stigma. And the bottom row are the aniline blue stains, so you can see all these pollen tubes growing down the stigma and into the style. We see the same image for RKF delta and RKF delta 2. So these pistils are supporting wild type levels of pollen tube growth when we've got Columbia pollen on these pistils. Now, the Circ1 BAC1 mutant pistils are not as successful in supporting wild-type pollen tube growth, and we noticed that there was reduced pollen tube growth into the stigma. And when we combine RKF delta with Circ1 BAC1, it was further exacerbated. You see very few pollen tubes entering the stigma. So I know this is a bit difficult to see, and so that's why we turn to the next assay, where we use these Gus-marked pollen grains as a way to track pollen tube growth. And so these are now looking at 2 hours and 6 hours post-pollination, and you can see the pollen tubes all stained blue when we use the Gus assay. So you can see, and we've drawn these dotted lines to show you where the pollen tubes are reaching as they're growing down the pistil. So you can see that Columbia, the RKF delta mutants basically look pretty similar. It's a little bit shorter growth in the Circ1 BAC1 mutant pistil, and even shorter in the hextuple mutant with RKF delta, Circ1, and BAC1 knocked out. So remember, this is 6 receptor kinase genes that have been knocked out. We can take measurements from here, and this is what the graph looks like here. So the bottom row of data is a 2-hour time point, so how far Columbia pollen tubes grow in Columbia pistils and the mutant pistils, and you can see that it's really all the same. But we see the differences at 6 hours. So the top row are the 6-hour measurements for Columbia pollen tubes in these different pistils. And you can see for Columbia, RKF delta 1 and RKF delta 2, they all support the same level of Columbia pollen tube growth. But when these pollen grains are added onto the Circ1 BAC1 mutant, you can see there's a decrease, so these wild-type pollen tubes are not growing as far. And when you then add them to the RKF delta Circ1 BAC1 hextuple mutant, we see a further decrease. So this is really interesting because we know that the pollen tubes were able to achieve wild-type levels of growth on the RKF delta mutants. But when RKF delta 1 was combined with Circ1 and BAC1, we got a further decrease compared to Circ1 BAC1 mutant. So this told us that RKF delta is functioning synergistically with Circ1 and BAC1 to support pollen tube growth. So the RKF cluster along with Circ1 and BAC1 are both needed to support wild-type pollen tube growth. Okay, so this is basically a nice summary of the work that we published. So we showed that the RKF cluster is involved in controlling pollen hydration, and the RKF cluster along with Circ1 and Circ3 support pollen tube growth. So where are we going from here in this project? Well, one of the questions we asked is what happens if we knock out more of these LR8-2 members? Will we see stronger phenotypes? And so I mentioned at the beginning that there's these other members in LR8-2 which show good expression in the stigma. These are the promoter Gus studies showing that there's strong Gus activity in the upper part of the pistil. And so we focused on knocking these genes out. And so far we've knocked out 3 of them out, LIC1, NLLR2, and RIR1, and combined them with our other mutants. And this is all the different combinations that were made. I just want to point out a couple of them. This bottom set in blue, these are only 8-2 members knocked out. So there's 7 of them knocked out. And this is the pistil highlighted in blue here. And just above that is our highest order mutant. We've got 9 genes knocked out. So it's these 7 LR8-2 combined with Circ1 BAC1 being knocked out. And that's what this pistil looks like here. So you can see that we're still producing pistils that are wild type in appearance as we knock out additional receptor kinase genes. And we then looked at the pollen hydration assay again, and what we saw was really interesting because what you'll notice if you just take a quick glance is that these are the wild type levels at 10 minutes, and then you get this decrease, but it plateaus out. And so previously I showed you that wild type Columbia pollen shows normal levels of hydration on Circ1 BAC1, but it reduced its hydration levels at 10 minutes on the RKF delta mutants. And what you can see is that as we add additional mutant combinations, the 7-8-2 members being knocked out, as well as Circ1, BAC1 being added in different combination, there's no change. So this tells us that the RKF cluster genes are alone responsible for regulating pollen hydration in the stigma, and these additional members are not involved. Next, some aniline blue stains. We saw a really interesting phenotype come up with these aniline blue stains. So we did see further reductions in pollen tube growth, but I'm going to show you that a little bit later. And what I'm showing you here is that we saw that the pollen tubes also seem to have smaller callose plugs in these higher order mutants. Okay, so these are wild-type Columbia pollen tubes growing through Columbia or mutant pistils. And you can see these little bright spots. These are callus plugs that are being deposited by the pollen tubes. So the reason why the pollen tubes are depositing these callus plugs is as the pollen tube is growing down the pistil, it needs to separate the growing active tip from the older parts of the pollen tube as a way to really concentrate the active cytoplasm to the tip the growing pollen tube trip. And so these callus plugs are laid down periodically to basically keep the active part of the pollen tube towards the growing tip. And so you can see in Columbia that these callus plugs are more elongated in shape. And so for whatever reason, this is in the 8-2 septuple mutant, and this is in the non-upple mutant. They have these rounder, smaller callus plugs. And when you measure the area of these callus plugs, there is a significant decrease on the Columbia pollen tube callus plugs when they're growing through these mutant pistils compared to Columbia. Okay, so we have no idea why this is occurring. We've never seen a phenotype like this in all our different studies. What I mentioned before is at this point, as these pollen tubes are growing through the stigma and style, that they're growing between the cells of these tissues, and the pollen tube is reliant in the reproductive tract for providing nutrients. So we're wondering if knocking out those receptor kinases is somehow impairing nutrient uptake, which is then impacting the callus deposition. And so at this point, it's a really interesting phenotype, but we're not really sure why it's occurring. Okay, so we also see reduced pollen tube growth, and this did become more significant as we knocked out additional members. So if you remember when we knocked out Circ1, BAC1, there was a decrease in the length of the wild-type pollen tubes compared to Columbia and the RKF delta mutants, and this was at 6 hours. And then when we combined them with Circ1, BAC1, we saw a further decrease in the pollen tube length. So there's a couple of data sets I want to show on here. So one is this box that I've highlighted here is the 8-2 septuple mutant. So it does not carry the Circ1, BAC1 mutations. It's only the 8-2 members, the 7 receptor kinase genes that have been knocked out. And you can see that at 6 hours, there is a significant decrease compared to Columbia and the RKF delta mutants. So this tells us that these 8-2 members on their own are required to support wild-type pollen tube growth, and when you knock them out, they're unable to grow as long through these mutant pistils. Then this last data point here is the non-upple mutant, which has the 7 8-2 receptor kinase genes knocked out along with Circ1 and BAC1, also known as Circ3. And now at 2 hours, we start to see a significant decrease in the length of the wild-type pollen tubes. So when we combine them all together, there's a further decrease in the ability of these pollen tubes to grow through these mutant pistils. Okay, so when you're impairing pollen tube growth, you could potentially be affecting seed set, because if the pollen tubes aren't reaching the ovules to fertilize them, then you're not going to get seeds being produced. And so we looked at how the decreased pollen tube growth impacted seed set. And on the right here is a graph showing the number of seeds per silique when Columbia pollen was used to pollinate Columbia pistils and the different mutant pistils. So you can see that Columbia and the RKF Delta 1 and 2, as well as the septuple 8-2 mutant, all have wild-type levels of seed set when they're pollinated with Columbia pollen. And then we see a decline after that. We have to always check when we see a decline to make sure that this is not just about ovule numbers, because if you have fewer ovules, that will also lead to reduced seed set. And so on the left here is a graph measuring the number of ovules per pistils. And what you'll see is that when you have the Circ1 BAC1 mutation, it actually introduces a reduction in the number of ovules per pistil. And this corresponds to the same decrease in the number of seeds per silique we saw when the Circ1 BAC1 mutant was pollinated with Columbia pollen. You'll notice that when we knock out the 7-8-2 receptor kinase genes, it does not affect ovule growth or ovule numbers. They're the same as wild-type Columbia. And so if you look at the seed set on the right here, you can see that there's a further decline in seed set compared to Circ1 BAC1 that's not related to ovule numbers. So this tells us that we are impacting the number of seeds that are being fertilized when you knock out additional members of these receptor kinase groups. And when you look at these siliques, you can actually see ovules that are not fertilized, which confirms what we thought, which was that when you have pollen tubes not growing as efficiently, they're failing to fertilize all the ovules. Okay, so we now can add more phenotypes that we've discovered as we've knocked out additional members of these two groups. So we've got the pollen hydration phenotype, the pollen tube growth phenotype, and now we also have the size of the callus plugs that are being deposited when these different receptor kinase genes are knocked out. So all in all, they're required for controlling pollen hydration and supporting pollen tube growth as well as callus deposition. So in the last couple of slides, I'm just going to talk quickly about one other direction that we're pursuing, and that is to look at whether these LR8-2 members might be involved in preventing interspecies pollinations. So this is an area that's recently cropped up for other players that are involved in compatible pollen pistil interactions, where they are also playing a role in discriminating against pollen from other Brassicaceae species. So I'm just going to show you one example of this. And so this is, these are aniline blue images where we've taken Capsella rubella pollen to pollinate these different pistils. So Capsella rubella is closely related to Arabidopsis thaliana, but we know from previous research that it will reject Capsella rubella pollen. So this is a Columbia pistil that's pollinated with the Capsella rubella pollen, and you can see that the pollen tubes have only grown a short distance into the stigma and then they've stopped. So this is at 8 hours post-pollination, and if you were, for example, to have Columbia pollen, it would be well on its way to have been fertilizing all the different ovules. And so this is with a Capsella rubella pistil that's been self-pollinated looks like. You have lots of pollen tubes growing down the pistil. When we added Capsella rubella pollen to our LR8-2 mutant, so with the 7 receptor kinase genes knocked out, you can see that these pistils are not as effective at blocking these pollen tubes, and they're actually growing a longer distance compared to Columbia. So there suggests that there may be some role for these receptor kinases in regulating interspecies pollen tubes as well. And this is something that we're following up on more now. Okay, so my final slide is to just put our data in the context of the models that I've already shown you. So I talked about Fronia and Anja having this negative regulatory loop to prevent pollen hydration, which compatible pollen has to overcome. And now I'm going to propose that the RKF cluster receptor kinases are also involved in this process because they're required for compatible pollen hydration, and so they may present a positive regulatory loop that promotes pollen hydration as part of this process. I've also talked about how these two groups of receptor kinases are involved in regulating pollen tube growth. So this is wild-type pollen tubes growing through the different mutant pistils were affected, and so I'm going to propose that they are involved at this stage here. And so we have the RKF cluster, LIC1, NILR2, RI1, SERC1, and SERC3 all present in the tissues of the stigma and the style. And somehow they're having a dialogue with this pollen tube to support pollen tube growth through these to this section of the pistil. And so these receptor kinase genes these receptor kinases are required for pollen tube growth and supporting callus plug deposition, and perhaps they may also play some role in blocking interspecies pollen tubes. Okay, and so to acknowledge the people who've done this work, well, essentially it's basically Francis Lee, who I've noticed has joined in on this webinar. Is a very talented PhD student who's off doing a postdoc in Switzerland now, and he basically powered through this whole project and produced this really exciting data that I've been able to present to you today. And I now have two new students, Laura and Stephen, who are continuing on this project and looking at the interspecies pollinations as well as some of the other phenotypes that I've mentioned here, and also looking for downstream interactors as potential signaling proteins in their pathways. I'd like to thank several groups for sharing resources with us in terms of plasmids for the BIS-C and expression data sets, and the ABRC where we got several tDNA mutant lines from. And to acknowledge my funding source, NSERC and KJEF for facilities support. And thank you to everyone for listening. I'd be happy to take questions.
Thank you, Daphne, for this brilliant talk. So if anyone has questions, I think the easiest way is to use the chat function and to ask your question just by typing it in the chat. And again, I encourage students and postdocs to ask questions first. Maybe while someone is typing, I may ask, yeah, maybe a bit of a, I don't know, stupid question, but you talked about pollen hydration. So how does the water get into the pollen? Where does it come from? Is it from the stigmatic papillae?
Yeah, so that's a really good question. And so it is coming, we know that it's coming from the stigmatic papillae, but how it's getting to the pollen, we don't know. We don't know whether there's aquaporins that are involved or other means in which the water is being released, but we know, yeah, so that's something that we're also interested in. There is some evidence, there's been some recent publications suggesting that there are some aquaporin type proteins that are involved that are required for pollen hydration, yeah, and they're in the stigma, yeah.
Right, thank you. So if there are no questions, I suppose I just keep asking because I have a couple of questions. So so those receptor kinases you mentioned, I think at one point you said they're like, oh, there's a question from Claire. Maybe we go with this one first. Do you have any hypothesis about the function of the pseudokinase DKN1?
Right, and so yeah, so I mentioned it's a bit of a complicated story, so BKN1 actually has in Arabidopsis thaliana, it's actually being knocked out in most of the ecotypes, and so there's very few ecotypes that actually encode a full-length BCAN1 protein. And we did actually explore one of these ecotypes, HH, and my student Betty, listed here, was able to knock it out, and we didn't really see much of a difference. So in terms of pollen pistil, pollen stigma interactions. And so that didn't really help us. Now, as you can see from my talk, that it was Arabidopsis lyrata BCAN1 that was able to interact with these receptor kinases. And so one of the things I would like to do at some point, and we need to try and get back to it, is actually try and use CRISPR to knock it out in Arabidopsis lyrata so that we can really explore its function. Now, some of the related members, so there's another related member, BCAN2, which has been implicated in plant defense responses. So it could be that we're looking at the wrong role for BCAN1, but there's a very interesting overlap between, for example, Feronia, which was first discovered as a receptor kinase regulating pollen pistil interactions, where it's required in the pistil, as you know, in a couple of different roles, is also a well-known regulator of plant immune responses. And so if BCAN1 has a similar function, it's possible that its interaction is more related to immunity, because the pistil, not only does it have to accept compatible pollen, it needs to keep pathogens out. So that's another potential hypothesis that we would need to explore. But at this point, we don't have a biological function for BCAN1 yet.
Thank you. So you mentioned that there are loads of those receptor kinases involved in this process. Is there a developmental or evolutionary explanation as to why so many genes are contributing to this?
So I think that, you know, it's sort of, I would say sort of, it's often the functional redundancy that you see in Arabidopsis thaliana is often explained that we're not really looking at the nuances. And I think that's true to a certain extent. So if we go back to this diagram here, we know I've mentioned a number of receptor kinases that are involved in the growing pollen tube tip, but there is recent work that suggests that PRK6 is the primary leader in this role. And so it may be that we're kind of missing the nuances as to why, you know, we're having to knock out so many to see a particular function. So.
And this cluster you mutated using CRISPR, did this originate by gene duplication?
It looks like it, yeah, because they're definitely the most closely related. If you look at in the phylogeny, RKF1 and the RKFL1, 2, and 3 are in a tandem array, and they also are most closely related in the phylogeny. So it would make sense that they're through tandem duplication that it's occurred. So.
Okay, so there's a question from Noni Franklin Tong. Does everything happen in the pistil at the hydration stage, or are there responses in the pollen at this stage?
Certainly people are starting to find mutants on the pollen side that affect these early stages as well. And but I think that now overcoming pollen, this hydration step, is not enough. Because, as you know, as this example, if I go back to the interspecies pollination, it's clear that, and we've also tested Eutrema salsaginaeum, that these related species are able to hydrate. So they're clearly able to overcome that initial step. And now the pollen tubes are being blocked. And so there's additional players that are involved in regulating these steps. I don't know if Nani, if that answered your question or not. So.
So as you were talking about those interspecies relationships, to which extent is this entire mechanism conserved in flowering plants? Is it always receptor kinases that are important for guiding the pollen tube?
Yeah, so I think people haven't looked that much into the other species, except for the work on the PRKs and the lures. So this is work that was originally done in, no, I'm drawing a complete back. I'm sorry, my mind's just gone completely blank on the species that it was originally, beautiful work done by um, uh, to identify the lures, and it was done in another species, which Tyrinia, which was and that was instrumental to actually leading to the discoveries in Arabidopsis as well. So
I see there are more questions here. Yeah. Tony has
another question.
Sorry, you go ahead. Yeah.
Okay, so how many RLKs do you think will be involved in the end, and how do you think the recruitment of these LRs have evolved? I have no idea. We're definitely getting a long list of receptor kinases, but I think the thing about plants is that they've clearly evolved receptor kinases as a mechanism for cell-cell communication, compared to, for example, animal systems, where the number of receptor kinases found in plant genomes is far larger than what you find in animal genomes. And so it really is one of the primary mechanisms of regulating cell-cell interactions. And these LR receptor kinases, a lot of them seem to have roles in plant immunity as well as development. And so how they've kind of evolved into all those different roles, I'm not really sure, but it may be that we'll sort of see these kind of, well, obviously the circs that we identified in this system are also involved in plant immunity. So whether the two are related, I don't know.
I have a technical question. You showed a number of beautiful images of pollen tubes growing in the pistil. So how technically demanding are those analyses? Is it difficult?
Yeah, well, I would say that the biggest learning curve when I have new students joining my lab is to just learn how to emasculate and pollinate these tiny little Arabidopsis pistils, and essentially not damage them in the process. And so I always encourage them to just practice until they get better at it. And that seems to be the biggest roadblock. Once you're able to do that successfully, then the aniline blue staining and the pollen gus assays, they're pretty straightforward. So, yeah.
Okay. Well, do you have any tips on not to damage such small and delicate parts?
Yeah, so be young and have good eyes, I think, is part of it. I don't know if I could do it anymore, so in steady hands. But yes, I think that you really do have to be very careful. People do have different tricks. For the most part, my students prefer to just use the anthers that have, I think it's important to have a fresh anther that's just a hist. If they get too old, the pollen may not come off as easily, and you might damage it trying to pollinate the pistil. And just be very light in your pollination. So, if you look at a naturally pollinated Arabidopsis pistil, it doesn't have a lot of pollen on it. So you certainly don't have to sit there and just, cover it with a lot of pollen. Just want to do a light touch. Some people like to use small paintbrushes. Some people like to use eyelashes. There's different ways, but it really is about the pollen application, I think, is the trickiest part, is that you get a nice pollination without damaging it. And typically, if you have been a bit rough with the pistil, you'll see all sorts of callus deposits in the papillae as a sign that you've damaged it.
Okay. Thank you, Daphne. for this interesting discussion and for this brilliant talk. I don't see any further questions, so I think we're coming to an end. I'm actually not entirely sure how to close the webinar. Maybe Rebecca can do that.
Okay, well, thank you very much, everyone, for coming and listening to me. I hope you
Okay.
enjoyed the talk today. So.
Okay. Thank you very much.
Okay.
And
Bye-bye.
goodbye.